To start with, I would like to wish everybody a very good morning to this special ceremony, this special academic ceremony, which I officially like to open, in which Ms. Snihirova will defend her academic thesis entitled Same Copy, Different Story, Environmental Modifiers in CNF Carriers. May I invite you, Ms. Candidate, to present a summary of your study and the conclusions of your thesis in the next 50 minutes, and I give you the word.
Thank you, Prorector. Dear Defense Committee, supervisors, colleagues, friends and family, in the next 15 minutes, I will present you the work that I did in the last 4 years in my PhD titled Same Copy, Different Story, Environmental Modifiers in CNV Carriers. Nature versus nurture. We all wonder, especially when we become parents, how much of human being is shaped by their genes and how much by the world around them. In science, this question matters even more when we study conditions with a clear genetic root. If genes establish a groundwork, what role does the environment play? This was the central focus of my work. To introduce genetics, our DNA is like a long text. Sometimes a piece is missing. That's called a deletion. Sometimes a piece is repeated. That's called a duplication. These changes are called copy number variants. Most CNVs are regular ones, so they're harmless, but changes in certain places can affect health. My research asks, beyond the CNV itself, what part of someone's environment, lifestyle, socioeconomic status, stress, change how the person functions? And how can we use that knowledge to support them? Most work on CNV conditions look at genes and the clinical pictures. Far less is researched about the environment. Yet daily context, family, school, healthcare, money, neighborhood, shape how people grow and learn. My thesis turns the spotlight on environmental factors and test which one tend to associate with better cognitive estimates in CNV carriers. As I said, most CNV research still centers on genes and clinical side, but not on the environment. And that matters because it may explain why people with the same CNV can have very different outcomes, from health to daily functioning. To make sense of this variation, I propose a simple two-layer model. A pathogenic CNV is a strong first hit. It changes how much of a certain genetic information a person has. But this alone does not determine the future of carriers. Two other layers help to explain why people with the same CNV can differ so much. First, the rest of their genome matters. Many small common DNA differences can adjust the activity of genes up and down and so soften or sharpen the CNV effect. Second, life circumstances matter. Supportive families, good schooling, stable income, and healthy habits can buffer the risk, while stress, disadvantage, or poor access to care can make problems worse, in theory. Put simply, the CNV sets the baseline, common genetic changes adjust the signal, and the environment turns the volume up or down. To study how CNV carriers respond to their environment, we need a clear expectation, what kind of sensitivity we might see. Each of these models predicts a different pattern of response to stress and support. By checking which model fits the result best, we can decide whether CNV carriers are mainly vulnerable to adversity, sensitive to both bad and good context, or especially able to benefit from positive condition. First model is being sensitive to negative events, meaning that the carriers are doing no worse or better under good circumstances, but they worsen under bad circumstances. Second model is sensitive to both. In harsh environments, they do worse than others, but in supportive ones, they do better than others, so sensitive to negative and positive. And the last one is sensitive to positive events only. They do not act worse than others under stress, but they gain more than others from supportive environments, so sensitive to positive only. In my work, we study how CNV carriers respond to their environments in 4 steps. First, we characterize the common variant background in regions where CNV usually happen. I need to mention that in practice, CNV happen in certain places, but exact breakpoints inside of CNV can change. In this first step, We study what happens in the whole area where CNV might occur, specifically concentrating on common variants in those regions. Then we analyzed large populational data to see how environment linked to outcomes in carriers of CNV, no matter whether they have symptoms or not. And next, we review the published evidence on environmental factors in mental health in one of the most common CNV-related disorder. And finally, we tested these ideas in clinical CNV samples. So these were the aims of this work. So research the environmental measures in CNV carriers in the populational and clinical data, evaluate how the environment influences IQ in CNV carriers, including change in IQ over time, provide ideas on how to guide personalized support, and set the future direction for the topic. Let us start from chapter 2. We ask a simple question. In the 8 genome regions where these CNVs sit, do the everyday DNA differences, common variants, relate to key traits and diseases, and how much? Here we use the data from big studies on everyday DNA differences for 20 traits, studies that collected data from regular people, not carriers of certain CNVs. First, we use the tools that group signal by region and test whether those regions show links to 20 traits. And then we used a second tool to get a size of that signal, how enriched those regions are compared to the rest of the genome. In short, we saw the 2 regions stood up. They show the most linked to traits and the strongest enrichment. We also observed 2 patterns. Sometimes the whole region carried the signal, like in region A, so it had the higher signal than any other individual gene in that region. Sometimes a few specific genes drove it, region B on the slide. This demonstrates that utilizing common DNA differences within CNV regions help explain why traits differ between individuals and help overcome the limitation of small sample size in rare disease research, as we utilize commonly available data on hundreds of individuals. In chapter 3, we ask whether the environmental matters for CNV carriers' cognition and well-being. In this chapter, we took information for populational data. We studied several well-known CNVs and 3 key environments: socio-economical status, stress, and lifestyle. We looked at 2 outcomes: problem-solving ability and well-being. Using populational data that includes people who carry these CNVs, with or without symptoms, we tested whether the gap between carriers and non-carriers become wider or narrower, depending on these environments. We found these patterns dependent on CNV for all groups. Overall, the effects were CNV-specific and different. However, our conclusions are very cautious because there were far fewer carriers than non-carriers. In chapter 5, we worked with clinical data. We pulled data from 3 European cohorts that use data on most common CNV-related disorder, 22q11 deletion syndrome, and followed participants over time. We asked whether everyday factors such as social-economical status, stress, substance use, sleep relate to IQ levels and changes in IQ. The goal was to pinpoint modifiable influences that might help support better cognitive outcomes in 22q11 deletion syndrome. We observed a clear linear decline over time in full-scale IQ, performance IQ, and verbal IQ. Parental education, especially fathers, was significant for IQ measures, pinpointing to a socioeconomical influence on cognitive differences in 22q11 deletion syndrome. Other environmental effects were less consistent, but the use of IQ change revealed changes that a simple cross-sectional design might miss. In chapter 6, we examined how environment relates to cognition in carriers of CNV in 2 regions, 16p11 and 22q11, and we looked at both duplication and deletion carriers in those regions. We took data from more than 1,000 carriers across 7 international centers and studied socioeconomical status, sleep quality, substance use, family environment, and stress exposure, and their influence on full-scale IQ, performance IQ, and verbal IQ. We used a specific approach also, an exposome-wide approach to compare carriers' profile and test which factors best explained cognitive differences. So in simple terms, we cast a wide net across many everyday factors, education, sleep, income, etc., and testing against IQ each of them with the same basic adjustment, and then retested the promising ones together to see which truly stand out as the independent explanation on cognitive differences. We have observed that carriers groups differed clearly in social-economical status and showed distinct sleep and substance use patterns. Across all CNV types, social-economical status was most strongly linked to IQ. Higher parental and household resources predicted better scores, and educational attainment was the strongest single predictor across IQ domains, with home ownership, parental occupation, and household income also contributing. And this current finding highlights the central role of socioeconomical factors in shaping cognition in pathogenic CNV carriers. So in this thesis, methodologically, we introduce a new approach using common variance data and populational data set to study CNVs and their link to complex trait and environmental measures. Conceptually, we highlighted the importance of considering environmental factors alongside genetics and pathology in CNV studies. And clinically, this work highlights the importance of collecting more detailed socioeconomical status information from carriers and encourage clinicians to actively support and promote improvements in the socioeconomical circumstances of families of CNV carriers. Overall, our results most consistently support the sensitive to both model. Many CNV carriers appear more responsive to both supportive and adverse circumstances. We also see some signs of sensitive to positive only or to negative only. However, to make sure that we see the clear difference between these models, we need longitudinal design and also moderate level of certain environmental factors that was not always possible in our research. For the field, the next steps would be first moving from single CNV views to network, genes, or network CNV. Equally important is globalizing CNV research beyond Western Europe and North America, building cohorts in Latin America, Africa, South and Southeast Asia, and Eastern Europe will improve generalizability and reveal context-specific modifiers. Next, we can use multi-omics model integrating different kinds of data, genomics, transcriptomics, epigenetics, and neuroimaging, and analyze them possibly using machine learning methods. We should standardize environmental measures with shared questionnaires and harmonized scoring, enable open, privacy-safe data sharing, and adopt longitudinal design with repeated, comparable time points and multi-informant input from teachers, peers, siblings, plus sibling-matched controls if possible. Bigger collaboration and including rare CNV might improve the overall understanding of how CNV work and improve the life and well-being of CNV carriers. And with this, I'm giving the word back to the prorector.
Thank you very much for your presentation, clear presentation, well in time, which gives us some extra time to ask some questions to you. So that's always appreciated. To start with, I would like to introduce several members who are here present. First of all, myself, I'm Professor Steinbusch, I'm the prorector, and I'm pleased that I will be assisted today by Professor Harwood. But more importantly is, of course, the promotion team sitting opposite of me. And we have 3 members in the promotion team, and I start from my right, so closest to you in the audience. We have here Dr. van der Meer, who joins us today, Professor Linden, who joins us today, and Professor van Amersfoort, who joins us also today. This is the promotion team who has guided the candidate for the last 4 years so that she can present her work today. And it's basically today that you're going to defend. We have 6 opponents for the audience. 5 are present here on site, and there will be 1 opponent online who will join us in the second place. So without further ado, we have still exactly on time. We're going to start with the opposition, and I would like to give the first word to Professor Rutte. He was the chair of the assessment committee, and he's affiliated with the Department of Psychiatry at our university, and I'd like to give him the word.
Thank you, Mr. Prorector. Dear candidate, first of all, my compliments to you. This is a wonderful thesis. I've read it with great interest, and it's really on a very exciting topic with fundamental questions that are being addressed by you and also by your very excellent promotion team. I also was impressed by your personal trajectory and your CV section in your thesis. It was very interesting to read where you came from and how you're now standing here today. And I have several questions, but I would like to start with asking you some further explanation, and that's particularly on what you see on this slide and also on the cover of the thesis. Can you explain us a little bit what is there and why you put this as the cover of your thesis?
Highly esteemed opponents, thank you very much for your compliments to my work and for your nice words, and that you actually mentioned the cover. So the cover was made by my friend. She is also a biologist. So idea was to put here a game, which is our life. So in our life, we all start, and then we go, and along our way, we see different environmental circumstances, sometimes positive, sometimes negative. So on this cover, you might see different environmental measures and how we all go along this way. I wanted particularly to make sure that there will be no positive or negative ones, clearly. I mean, there will be no dead end, because we never know what is positive, what is negative in the end. So they're all factors, but it's just part of a path. But there is not going to be like something that will bring you back, because you never know which factor is exactly negative or positive. Also, some of them are related to my own life, which is dancing or meditation. So we wanted to make it a bit personal and show that the life is game, and put some environmental measures as a reference to my work in there as well.
Yeah, thank you very much. Very interesting. And I was also expecting something alike, also with the swamp of procrastination. Very interesting. So many thanks. So then to some questions about the content of your thesis. And this is more an overarching question, relates to chapters 2, 3, 5, and 6. In the sense that you also introduced the different theoretical models of gene-environment interplay, and also particularly you are talking about the diathesis stress model, differential susceptibility model, and the model of vantage sensitivity. And these all revolve around sensitivity to an environmental factor. Now, I was also thinking, now sometimes people with a certain genetic background are not only more sensitive to a certain environmental factor, but maybe also be more likely to exposed to a certain environmental exposure. So that relates to gene-environment correlations, if you know what I mean. I was wondering, have you incorporated thoughts on this topic in your thesis, and if so, how? I mean, you don't study gene-environment interaction per se, but you do this with carriers and non-carriers, and you look at prevalences and cross-sectionally. But how do you think about the gene-environment correlation as a one factor or one pathway towards increased risk?
So I can answer these questions in two parts. Firstly, there is a sort of limitation to my work, because majority of them was made based on European cohorts that all would have maybe better environmental circumstances than if I would make my research in other parts of the world. So I think the baseline is different than if I would make my research, let's say, like in Asia. So chapter 3 was based on British data. Chapter 5 and 6 was European cohorts. So first of all, we need to remember about that. And what about genetic and environmental correlation? Unfortunately, for some of the chapters that I did, we cannot get the genetic component because we didn't have information on that about, for example, IQ of parents. That would be put in the model and then try to extract the genetic component that would influence cognition in carriers, or that we cannot get non-transmittable polygenic risk score for education from the parents that we would also like to put into the model. So sometimes for certain data sets, we didn't have that information. I understand that, of course, we have environmental measures, but parental education also influence the children through genetic nurture. So they create better environment, and it's indirect influence, which is not neither environment, not genetic, but together. But to distinguish this element, we needed to have information on parental IQ also, and maybe some genetic information that we could put in the model, which we didn't have. That's why we mentioned it briefly in the discussion, but we did not go, we cannot put it into model, unfortunately, so we could not basically.
So I understand what you're trying to convey. I was thinking maybe it's too early to try to disentangle these effects, but I think it would be one explanation, one possible explanation for finding that you would have, that low socioeconomic status is more prevalent in carriers. It could be that carriers are more sensitive to this effect and then have increased clinical expression. Could also be that they are more likely to be exposed to socioeconomic status or end up in a poor socioeconomic class. So maybe that's something that we can address later on. And continuing on this line, I understand for practicality reasons you particularly focus on stress, socioeconomic status or class, and education. What was the third environmental factor?
Lifestyle in some chapters, sometimes sleep.
Oh yeah. Yeah, but sleep is that environmental, is it physiological measures? But I was wondering, in earlier life, because this is mostly what you focus on adult life, and of course with genetic syndromes, you see developmental developmental issues. I was also thinking maybe these individuals are much more likely to be exposed to birth or pregnancy complications or more sensitive to these. You didn't study these because probably these data were not available to you, but maybe they will become available. What would you think would be the most relevant early environmental factors that you would like to include in your models?
Yes, I think birth complication, we had this information, like there wasn't a data, but we did not include it in the model because there was not available for a lot of people. So it was a really small piece of information that would not give us a lot in terms of a model. So yes, I think birth complication is a nice factor to put. As for idea that these carriers have a worse socioeconomical status, I'm not sure because usually if we talk about 22q11 deletion, it's usually de novo, so it might happen in any family with any social economical status. Now, I might say that we might see different stress relationship in these carers because they are more, like the parents more predisposed to certain stress due to the condition of their children, and that might influence, but yeah.
Thank you very much, very clear. Great. So I would like to give back the word to the prorector.
Thank you very much. We will proceed online, and I would like to welcome Professor Skoes. He is affiliated with the Department of Developmental Neuropsychiatry at the University College of London, and I'd like to give him the opportunity to discuss with you. Professor Skoes.
Thank you very much. Thank you for inviting me, and I'm sorry I can't be there in person. This was a fascinating thesis. It was a subject that I'm particularly interested in because we've studied these CNVs in my research group for the past decade or so. And it's so difficult to try and differentiate the importance of environmental measures from other factors that are directly related to the genetic changes, as you've discussed. I wanted to ask you some questions about potential confoundings factors, because you talk about environmental influences, but differentiating those from a term that you've used, which I think is very appropriate, genetic nurture, is exceptionally difficult. When we talk about parental IQ or parental education, of course, the likelihood is that there is a relationship between those two things, that parents with higher IQ are likely to have higher education and provide ultimately, through socioeconomic factors, a more stimulating environment for their children. You've acknowledged that. So my question is about the heritability of these conditions that you've described. I think there were three conditions primarily. I'm focusing largely on chapter 6, but this does relate to a number of other chapters. There's the 22q11.2 deletion syndrome. There's the 16p11.2 duplication and deletion. The heritability of those three conditions is very different. It's not something that you've discussed, but could you tell us something about that?
Hi, esteemed opponent. Thank you very much for your words and for your compliments to my work. Yes, so as I said, usually, for example, if I know that 22q11 deletion is usually happening de novo, so it's not 90% of cases, if I remember correctly, so it's not usually inherited. Yes, correct. And for other CNVs, the rates is different. That's why we do have the type of, like, I'm not sure that we have information for everyone, whether it's inherited or not. We can put it in the model in the next revisions and discuss it in discussion. But there is a difficulty to distinguish, like we have heritability of a disorder itself and we have the heritability of like environmental factors that are not completely environmental. So that's why I absolutely agree that it's very hard to distinguish genetic and environmental in this case. But I think it doesn't matter that we don't need to make any research on that. So it doesn't matter that we should not try to still build a modern L and try to find certain candidate genes and pathways and not wait for perfect circumstances and perfect data set to happen when we have all this information and we can perfectly distinguish all these different factors like parental NIQ and education and polygenic risk score for IQ and child and polygenic risk score for IQ and education and parent, and then build this beautiful, perfect model. So we worked with what we had information and tried to set the direction for the future research as well, and to think what's happening in our data and what we can focus on, and also set for the future studies as well.
Okay, can I just draw your attention to something that you talk about in chapter 6, which is that those with the 22q11.2 deletion, parents of children with children who've got 22qs, parents have higher education and occupation scores, have higher IQ. Now that's not too surprising because, as you said, only 10% of that condition is likely to have been inherited. However, with the 16p11.2 duplications, you say there's a parental income and parental education are somewhat different. Actually, around 70% of that condition, as I understand it, are likely to be inherited. And of course, interestingly, you say that the parents of children with the duplication, which is the more heritable form of 16p11.2 CMVs, that's twice as likely to be inherited as the deletion. And interestingly, those with deletions have less intrafamilial conflict. My interpretation of that would be that because the parents are more likely to be carriers in the case of the duplication, that could well have a consequence on the environment in which the child's being raised, because there's more stress in those families. And indeed, that would fit with our own research, which I mentioned we've been doing over the last decade or so. So it is important, actually, to take into account the fact that these conditions are variably heritable in your model, I think, even if you don't know, of course, whether the parents were carriers or not. Another issue that you don't discuss, but I think is pretty important, is that given the very wide range of ages of the subjects in your study, some of whom were children, of course, but a great many of whom were adults, they will have been through the education system. I appreciate different education systems in different countries, but in the course of their education will presumably have received varying degrees of support at school because of their genetic vulnerability and their consequent phenotypes. Do you have any information at all about the environmental variable of those people's education, of your subject's education? You talk about intrafamilial variables, but you don't mention, as far as I recall, anything about variables that really must be very important, but occur outside the family.
So in chapter 6, if we're talking about it, yes, we had information of education level of the participants. And we have information on education level of parents, but I'm not exactly sure what exactly you want to ask related to that.
Oh, well, if you have a child with a genetic anomaly who's got learning disabilities, which of course most of these children will have, then schools provide extra support for that in varying degrees. It's a huge issue in our country as to whether parents can get that support or not. And indeed, parents who are themselves carriers are less able to get that support for their child. There's an interaction there. I'm speaking from research that we've done, some of which has only recently been published, so you wouldn't have seen it. But as a general point, I think it's you can't assume these children all had equivalent education. And obviously their ability to perform cognitive tasks subsequently will presumably be somewhat related to their own educational experiences, not just because kids spend most of their time at school actually during their childhood, apart from holidays. They're at school, so that's a big environmental impact on those children's adjustment and progress.
Maybe I'm wrong, but if I'm assuming that one of CNVs is inherited, and it's inherited from one of the parents, we still have education, and we have still another parent whose education might matter and was not influenced genetically by the CNV itself. So
I'm talking about the children's education.
Dr. Skoes, Dr. Skoes, Professor Skoes, I do have to interrupt because we have to proceed. The time is over already, and I think we have to proceed to the next opponent, which is Dr. Singskoks. And I would like to welcome her that she joins us today, all the way from Nijmegen, and she's affiliated with the Department of Genetics and Developmental Disorders, and I'd like to give her the word.
Thank you. So first of all, I would like to congratulate you, Dr. Snihirova, with this excellent thesis, and I would like to extend my congratulations to the supervising team, of course. And many studies have explored CMV syndromes, and you look particularly at biological aspects or genetic aspects or phenotypes, but studies exploring environmental aspects are rare, and that makes your work very innovative and relevant. I also very much appreciated that your propositions quote from two sacred books that are my personal favorite, the Bible and Harry Potter. And I particularly like Professor Dumbledore. But yeah, I would like to talk about Harry Potter, but we're here to talk about your own book. And so my first question, I would like one of your paronyms to read proposition 2.
For translating modifiable influences into personalized care for individuals with rare genomic disorders.
Thank you. So I'm a clinical psychiatrist and I work with children and adults with genetic syndromes. And as a clinician, I've read your thesis with interest because I was very keen to learn about environmental modifiers of CMV syndromes. Because we all know that CMV syndromes such as 22q have enormous impact on brain development, and there's really nothing we can do at this stage, to change that. But environmental factors might hold the key to modify outcomes potentially. This is very exciting. But after reading your thesis, I was a bit puzzled because I wasn't certain which modifiable environmental factor has the biggest impact. So my question is, based on your own work, what do you think is the environmental factor that I should focus on in my clinical work with individuals with 22q?
Highly esteemed opponent, thank you very much for your comments, and I'm excited to talk both about Harry Potter and my book, but yeah, let's talk about my book. Yes, as I saw through my chapters, socioeconomical status was prominent in many, many cases, but I understand it's very hard to change at certain points. However, I still think that even if you can influence education, like promotes courses or specific schools or personalized plans for children, even if it's already later in life, it still might benefit in the future. Same with some financial aid. I mean, I know that we are talking about very perfect world with rainbows, but I think we still need to think that if we see this being significant in like for me, for my studies, that I saw it significant in so many chapters, it's still possible to try to incorporate government and some policies to provide personalized support, even if it's a bit later in life.
Yes, no, I do very much appreciate that answer, because one of the things that struck me in your thesis is that SES is basically the driving factor behind lots of sort of more poorer outcomes. So this is all about health inequality and mental health inequality, and you mentioned quite rightly that, you know, funding better schools, more educational opportunities for these children with CMVs might really be helpful. And so I was thank you for that answer. I'll just move to another questions, because in chapter 4, you review all the evidence on environmental modifiers in 22Q. And you summarize this very nicely on page 81 of your thesis. And many of the environmental modifiers you identify for 22Q have also been identified as environmental risk factors for mental illness in general. And, well, for example, we know that higher stress loads is associated with increased risk of psychosis in the general population. And my question to you is, did you expect this? So did you expect to find sort of similar modifiers in the CMV population compared to the general population?
Yes, I think we expected that. That's why it was like in almost every chapter, but also it was, of course, based on data we had available. However, I need to mention that that's why I promote so much in my thesis, that we need to ask more questions from the patients and their families about that we were lacking data, because again, if all this research is concentrating on pathophysiology, we don't have much information about any life circumstances of these people. That's why we need more. And that's why there are not a big table on page 81, because we don't have many studies that, again, based on the information that we have. And majority of them is, yes, on stress and intelligence, but not on, for example, migration or urbanicity, or, yes, some of them on parental relationship, express emotions. But again, some information is still missing. And I know that we are talking about rare disorders, and we need to combine data from different data sets. That's why also important to collaborate more and ask same questions. You can ask one question about parental education in 10 different ways, and still it will make very hard work for statisticians and to analyze that.
Yeah, no, that's great. So more research is needed also in global, you know, across the world, really, because lots of what we call Western countries are involved now in the big consortia, but less so Africa and Latin America, maybe, yeah, or South America. So were there also environmental factors or modifiers different in the CMV population than in the general population that you came across, or was it mostly similar? Were, you know, the environmental factors mostly similar in CMV populations as in the general population in terms of mental health risk, or were there also differences?
You mean that their role in like cognition and mental state, or like what? Because I mean, we all exposed to environment, we all exposed to different, but our sensitivity is different depending on like genetics, for example. Or this, we try to compare and think about how CMV carriers differ from controls. In their perception of environment. And based on this chapter, we also saw that there are not so many between group studies, which we call that there are not many studies that actually compare 22Q to any other to control. So you need sibling match control, which was lacking. So some of the studies that I mentioned in within a group study was actually had control, but they never did any interaction. So they never compared them. They did separate analysis. Some of them tried to compare with autism spectrum disorder and intellectual disability, but that's why we need not just more data, but also more comparison between controls. There are not so many studies about that.
Yeah, no, that's right. If I'm correct, right.
Dr. Sixler, sorry, we also have to proceed, so I have to close this discussion too. We have 3 more to go. And thank you very much. And I would like to continue to Professor Harwood. He's affiliated with the Department of Translational Genomics at our university, and I give him the word.
Okay, thank you, Pro-Rector. I'd like to congratulate you on your thesis. I've enjoyed it very much, and I think it's incredibly relevant for today. So that's really good. And I'd also like to congratulate you on your fantastically simple but meaningful slides. So thank you. I thought that was very impressive. as you probably know, I like to ask questions about mechanism, okay, and at lots of levels. And I was very struck in reading what you presented was really about using IQ as a measure, which makes lots of sense. But I came away with the impression that what we're looking at is IQ being modified or changed by the conditions, which I guess is a question of how do they change. But actually, I would like to ask you a bit more about what is changing and why are those measures changing?
Hi, esteemed opponents. Thank you very much for your nice words about my work. Just to clarify, if you want to know what is changing, you mean in IQ? What is changing?
So IQ is a big measure, okay? And I know that you showed different versions of it. So what element of that measure is actually changing in the different conditions? And I'm going to ask you about aging, so maybe we could start thinking about chapter 5 and what is changing as the individuals of the cohorts age, which is what you showed.
So I think depending on the disorder, we have different areas of cognition that are mainly touched. So we know, for example, that 22q11 have verbal difficulties. That's why verbal IQ might be more impaired than any types of other IQ. So I think it's we are talking about problem solving, we are talking about areas, we are talking about verbal abilities, and so different ability to cope with different tasks.
Okay, that's in general.
Yes.
So a more specific question then, do you think that the changes you see in IQ between the different copy number variants are caused by the same changes, the same effects, the same mechanisms, or are they different from one CV to the next?
Yes, of course, because when we're talking about CNV, we're talking about deletion or duplication happening in certain region and boundaries, but we know exactly what genes are affected. So deletion, duplication might vary between the size of it and the genes involved. So of course, which genes are touched, involved in which processes, and it could be any like plasticity, development, synaptic transportation, that would lead to worse memory and like vice versa, affecting different pathways. So in ideal world, you need also to look what exactly areas, so whether it's a deletion like a CD region or AD, so also look at the exactly genes involved.
Okay, yeah, that's a very specific answer. But do you think, though, that the change in IQ is caused by the same change in every individual, or could it be different from one individual to the next? So could IQ be quite heterogeneous in its nature?
Quite heterogeneous because, again, plus environment, plus except for just deletion, we have other genomes that also matters. It could be difference in how you silence enhance genes, you have transcriptomics, you have so many different mechanisms happening in a cell. So I think it's also different. We don't know about the rest of the genome. We don't not only know about this deletion, but we not necessarily have like whole genome sequencing for each individual to find it. That's why I was also mentioning that we need machine learning to get all this data and analyze this all together.
Great, okay, thank you. And then just to go to the specific case of changing IQ with age, which maybe you could explain a bit more, and why do you think it changes?
So that we observed, yes, right, in chapter 5. So it was a decline in performance IQ with age. We did not find actually like that. I think there was a study that looked at that and they didn't find any significant decline in 22q11. We tried to speculate that it might be that nowadays the people with this disorder live longer, so we might see more degeneration because previously they would not live so much. But again, it just was an idea also about the Parkinson's disease involving, but that we found a bit in the literature, but we were not sure exactly why it's happening. And whether it's just age-related or it was just regular decline. Okay.
And just one follow-up question, if I've got a minute. And so the age group, the median age group, which is what's on that graph, changes from 13 to 16, I think, So degeneration could be one explanation, but could it also be the transition from childhood to adulthood, adolescence?
Yeah, probably, yes. Why? Yes, why not?
Right, okay.
Yes,
Right.
you're done.
Thank you, Prorector. Thank you.
Thank you very much. Then the opposition will be continued by Dr. Blokland. She's affiliated with the Department of Neuroimaging Genetics at the Institute for Mental Health and Neuroscience, and I'd like to give her the word.
Thank you, Prorector. Dear candidate, firstly, congratulations to you and your promotion team on delivering this very interesting thesis that I enjoyed reading. I would like to ask you a couple of questions on your mechanistic interpretation, and then, if there's time, a more general question of what's next. So my first question is about chapter 2, and in your discussion, you mentioned that you found a strong correlation between the number of associations and heritability, suggesting that the increased associations between CNVs and specific traits arise from a greater genetic influence on phenotypic variability rather than environmental factors. And you hypothesize, and you cite a study saying this may be attributed to distinct chromatin accessibility in the 16p regions, CNV regions, for instance, as shown in human brain tissue. I was wondering whether you can explain a bit more about what maybe this study found that you cited and how you made that connection to chromatin accessibility.
Hi, Listin Popanets. Thank you very much for your words and for your question. So yes, right. In this chapter, what we wanted to see is, except for linking the common variants in the regions to traits, we wanted to see whether the traits themselves exhibit high polygenicity, and also we checked heritability and discoverability. We were looking for traits with high polygenicity and low discoverability, mainly because then it would mean that they are influenced by many genes, but they're highly hard to detect, which we found, and we also checked heritability using mixer here. And since heritability is related to discoverability and polygenicity, basically it's a multiplier by heritability, if I remember correctly, results in discoverability multiplied by polygenicity, so they're opposite direction. So we here basically try to explain two different things. So in the first part is why number of associations with region was not correlated with polygenicity by heritability. So and try to explain that if these traits are highly heritable, that's why we might see so many associations with regions. And the second part was mainly to explain why 16p11 was connected to so many different traits at the same time. So if they have higher accessible genes in terms of that easier to get and transcribe the proteins from them, that means that they have higher active genes in there and connecting to multiple pathways involving many genes, and then why it might lead to connection to height, to mental disorders, anthropometrics, and so on. So basically I here try to describe two different things in one paragraph, which might maybe divided into two.
Okay, and chromatin accessibility, it's an epigenetic process, and it can be environmentally driven as well. So is there the potential that you say that it's likely not environmental factors in this case? Is there a potential to for these environmental factors to act through this chromatin accessibility directly?
Yes, as for this, we can perform H-magma, which is another test using high C methodology to see exactly what other SNPs not in the regions that also influence the association between trait and a region. That would be an additional sensitivity analysis to check for that, but what other SNPs in any other different regions have also influence of this association. And this is made exactly with this test of accessibility of where this region is located and how easy it is accessible. So for this, we need an additional analysis, and then we can explain it more.
All right, thank you. Then one general question, and this is related to the general discussion chapter. in which you give lots of suggestions for future research. And ideally, all of it would happen in the immediate future. But given that funding is limited, where do you think the priority should lie? What kind of research do you think will overall yield the most information and maybe make it possible to translate findings to clinical practice? In other words, where do you think we can get the most value for our money and why?
I think we need data. And from data we get research, and from research, I'm not sure exactly how financing works in terms of how you get money for your research. So from my point of view, we're talking about rare disorders, so we're talking about very rare, very limited data. So I think collaboration, that would be the first thing, like sharing data between the consortiums and countries, it would be the first step. So if you gather more information, then from them you can go to more analysis and more research and do more with that. But I'm a data person, that's my view, because I think incorporating machine learning is too much computational power and money, and I'm not sure whether clinicians have time to ask more questions, and whether it's actually in a protocol, if it's so easy to change it. Yeah, I think no.
Yes, all right, thank you. I'll leave it at that.
Yes. Thank you very much. Then the opposition will be continued by Dr. Harwood. She's affiliated and working in the field of psychiatric genetics, also at the School for Mental Health and Neuroscience, and I also give her the opportunity to discuss with you.
Thank you. So, dear candidate, I would also like to congratulate you on a really beautiful thesis. It's very interesting, very nice work, and very well written. And I did appreciate the well written. I'm going to ask you a few questions about the data. So in chapter 3, you used UK Biobank data with participants aged 40 to 69, with probably quite similar socio-economic status. And I know that Biobank is quite renowned for these features. But then in chapter 6, you had access to the genes to mental health data, which has a wider age range and probably, I think, wider socio-economic status. But maybe you could tell me about that. So I'm thinking, which I'm sure you are as well, could you merge the two data sets to give more power to your analysis if you were going to repeat any of the things going forward from your current work?
Hi, esteemed opponents. Thank you very much for your comments to my work. I would not merge this data, but because this is different types of data. So UKB is populational data, meaning that the people that we looked into were carriers of CNV, but not necessarily showing any symptoms. And clinical data is people who have clear symptoms, clear pathophysiology that we can see, and I think they're not comparable. We also assumed, and we mentioned this in our limitation, that in UKB, carriers are probably healthy individuals who volunteered for being part of UKB, and I'm not sure that we cannot merge them with clinical data sets. I think it could be compared and contrasted, but not merged.
Okay, thank you. So another question about data is the FAIR principles are a set of guidelines for data management that aim to make research data findable, accessible, interoperable and reusable. And I think you've referred to quite a few aspects of this throughout your thesis, and particularly at the end, but I would just like to know how you got on during your PhD in this aspects with finding data sets, and how fair was the data that you used, and do you think these aspects of data in general could be improved, and if so, which parts of that would you think would be most important?
Yes, that is right. The data that was collected, especially for Chapter 5, was from different sources, used completely different data initially and was not aligned, different scales, and that's why in analysis we had to cut a lot of data to analyze it. So something that had different levels, we had to binarize, get these scores, unfortunately, because it was not presented. Also, the problem with rare research diseases is that you cannot share all the data, especially environmental, because since it's rare, you can easily find a person if you know so much data about their economic status. So, for example, if you have Thomson Deprivation Index, which is UK index for neighborhood, you can figure out. So I think it's very also tricky to be very careful. You need to be very careful with the data, and especially with environmental data, to make sure that it's rightly protected. So my impression of the data was that it could be improved through collaboration, through same questionnaires across cohorts. That was Gene to Mental Health tried to do, and through more open source, but at the same time protected portals. So you cannot transfer it so easy through home laptops.
So federated.
Yes, exactly, and also be not dependent so much on politics.
That's a very good point, yes. Okay. And so the next question, what do you think is the most important finding in your thesis, and what impact do you think it could have for the participants with the CMV carriers?
I think for me the most important is the link between parental education and IQ, which emerged in both Chapter 5 and Chapter 6. For me, this was interesting, especially difference between mothers and fathers' education, which was interesting in terms of that would you expect that the father's education was more important in certain analysis than mother's education, which was interesting for me. As for 22q11 research, you still can promote education for parents even if they already have a child with 22q11 or any other CNV. Maybe it's not the best example of environment that is modifiable, but still it's promising. And
do you think that the difference between the mother's and father's education is explained by sort of Western society, and that if you looked in a different cohort you might find some differences, or do you think it is, and can you explain any of that?
We proposed that probably in the majority of cases, father's education has more correlation with socio-economical status of a family and income. That would lead to it, and mother's higher education maybe might mean that the mother spend less time with the child, and that might also influence IQ in different way, and have also higher stress of mothers because usually mothers is primary caregiver.
Mm-hmm. Okay, thank you. Do I have another minute, or are we? Yes, you are. Research opens up further questions always. So now that you've completed your PhD, where do you think the next PhD student should begin?
I would like to continue work on environmental and genes in CNV carriers and beyond. But particularly here, it would be good to finish Chapter 6 with more data available, and to add more information about if we had at some point sequencing data, we could try to get the genetic component of education and IQ from the picture, and to make sure that we actually see the environmental measures. So this thing would be the next steps my research.
Okay, thank you very much. I'll stop there.
And then the beadle is here right on time, as you noticed. Yes, thank you very much. So, Ms. Snihirova, the time appointed for defending the thesis has now officially passed. The degree committee will now withdraw to discuss not so much the quality of your thesis that has been approved, but we're going to the way you have defended your thesis, but also the way you have presented your thesis. I requested you and the company here, your company here, available in this audience, await the results of all our deliberations and the return to the order. Thank you very much. Dear Ms. Snihirova, the degree committee here present has assessed the quality of your thesis, but in particular, the way you have presented your thesis in the first 15 minutes and the way you have defended your thesis in the last 45 minutes. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor van Amersfoort is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and therefore I invite your supervisor now to take the floor.
Do you promise to work in accordance with the principle of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Yelyzaveta Snihirova, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
I would like to give the word to Professor Linden for a second qualification scholar. Yes.
On behalf of the Research Institute of Mental Health and Neuroscience, I'm also entitled to hand out this official certificate. The owner of this certificate has received training in a broad range of academic skills and has gained experience that is relevant for a successful career as an independent scholar. Congratulations on behalf of the Research Institute Men's.
Thank you.
And this will continue by Professor van Amersfoort, who will speak officially the laudatio.
Dear Lisa, dear Dr. Snihirova, congratulations. You did it. Well done. And I also would like to extend my congratulations to your family, who came especially from Ukraine to celebrate this wonderful milestone with you today. Looking back at your journey with us at the University of Maastricht, it started more than 4 years ago. In September 21, you came to us. You started as a full-time PhD student under supervision of Dennis, David and myself. We had obtained an internal grant for a PhD student on a project looking at the role of environmental factors on neurodevelopmental outcome in copy number variant carriers, using large population and clinical data sets. So we advertised a PhD position for this exciting project. You were selected and moved from abroad, where you were living at the time, to Maastricht to start your PhD trajectory. This all took place during a challenging time. We were still dealing with COVID-19 and in and out of lockdown measures. So this is not an easy time for a new PhD student. Hard to meet new people in a new place, in a new country, and integrate with the university's PhD culture, with communication often taking place via online meetings. From day one, you worked very hard and made a very motivated impression on us. We were particularly pleased that you were so keen on research into neurodevelopmental disorders related to CNVs. In your PhD thesis, which we've just heard this morning, you focused on the effect of environmental factors, including stress, socioeconomic status and lifestyle on clinical outcome. The field of CNVs and related clinical presentation is under-researched and under-serviced, and this is particularly true for environmental factors, which make this topic so important. We frequently hear from patients and their carers that they are very interested in the role of environmental risk and protective factor. So we expect in the future a considerable interest in your research and the publications you've written from the patient community. Another challenge you faced during your PhD trajectory, and this also became clear over the last 45 minutes a bit, that your trajectory included lengthy and difficult procedures in obtaining several data sets from collaborative parties, which data sets that are not always complete. And this was beyond your control and required a lot of patience and perseverance, particularly near the first half of your PhD. And for us as supervisors, we always had a plan B in mind in order to keep moving ahead towards a PhD thesis that could be ready in time, which we managed to do together. In preparation for today, I reread your application from 21. And what is clear to me, that as I got to know you, is that you're a very curious and eager learner. Your CV is characterized by a diverse background in neuroscience, genomics, computational neuroscience, medicinal chemistry, and biophysics. You applied for a PhD position because you wanted to apply all these diverse skills to psychiatric disorder. At the time of your application for a PhD position, you were studying in Berlin and Amsterdam as a Erasmus student. I had never heard of it, but it's a prestigious European Neuroscience Master Fellowship program funded by the European Union. You already then liked the Netherlands, and I quote from your application letter. After 1 year living in Amsterdam, I fell in love with the Netherlands. I will not exaggerate if I say that this is the best country in Europe for working or studying, especially as an international student. Almost everyone speaks English, there are plenty of opportunities for sport, and the country is generally very cozy. Safe bike paths everywhere, and the most delicious ice cream I've ever tried, Jumbo Roomijs Choco Banaan. And the crying seer girls in the mornings. So, you gave a personal touch to your application letter. There was also in that application letter some self-reflection when you describe yourself. And I must say, that was spot on. Now I got to know you and see what has happened since then. I got a strong belief in my abilities to adapt to anything and be able to follow up in my plans despite unexpected obstacles. Well, we've already mentioned some obstacles, but little did you know back then which other unexpected obstacles were ahead of you. Almost 6 months after the start of your PhD, when the pandemic was finally gone, you were faced with the horror of the Russian invasion into your home country, where your family and friends, whom you had left 6 months earlier, had to live their lives in daily fear. Although you were very worried, obviously, you never showed this to us and focused uninterruptedly on your PhD work. You managed to host your mother and grandmother in your little flat in Maastricht. Unfortunately today, almost 4 years later, the war is still ongoing, and over the past years you regularly went back to Kiev to see your family, sometimes under very difficult circumstances. I hope you felt support from our men's PhD community over the last years. We have got to know you as a very social person, active as a PhD rep for Division 2. You've organized several social activities with other PhD students, and in addition you helped organizing events such as the Men's Research Day, the weekly LAM meetings, and the Division 2 Day Out. And recently you got a new job as a men's photographer for educational and social activities. Despite all these challenges, both in and outside your PhD life, you finished your thesis perfectly on time, which is very, very impressive. As a postdoc, you will continue to work with us on the gene to mental health dataset and also work with the CMV imaging datasets we have. You will go beyond the techniques you used during your PhD and develop and evaluate multimodal machine learning models that classify the different CMV groups. This is also a field in which international collaboration is of particular importance. And during the past 4 years, you liaised very well with our international partners in Belgium, the UK, the US, Canada, and other countries through the Genes to Mental Health and Minds collaborations. Dear Lisa, we hope that these links with international partners continue to open new perspectives for you and will prepare you for your further international research career. But first today, you celebrate this big achievement today together with your family.
So, dear Dr. Snihirova, I would like to also congratulate you in your new title, officially. Also on behalf of Maastricht University, I can tell you we are very pleased. Also the presentation which you gave was clear, what we consider for a lay public, and it was really for a lay public. I like to compliment you. The answers the way you responded to all the answers here of the committee were very adequate, and I can tell you that therefore we came to a unanimous positive decision, so there was no doubt about your thesis. In my congratulations, I would like to have also bring forward the family. As I now understand, they came all the way from the Ukraine. I think it's a long flight or a long drive. I'm not sure how you came, but that we discuss later. I must personally say that I liked your thesis too. It was already mentioned by the first opponent about the front page. Well, I can tell you, I used the front page as well, because I played it a lot with my granddaughter, and we had all kind of communications about what actually was on the front page in the different subjects, like you have over there. She has to explain to me what the ladder was, the forest was, the children were, and we played a lot about this. It was nice. Saying this, I think we come to a conclusion, and I would like to mention here to the people in the audience, there will be a reception on site. We will come later. First, we make here a picture with the new young doctor in the aula, together with Dr. Skoes. We will be online. Thereafter, we're going to make a picture on the stairs of our university, and I would also like to invite the family to join us later, so that she can be on the picture as well. And thereafter, we're going to meet you, and it will be in approximately 7 minutes. That's the time which we have scheduled for this. So I hope to see you there, and by this, I'm going to close officially the ceremony in which I use my hammer, which I have for you. And this is officially closed. Thank you.